pleasure to welcome you all to Lindsay Dade Defense. Lindsay came to us. She was actually living in St. Thomas already when she started the master's program. She'd been here for a couple of years, I think a couple of years, as a scuba instructor at Koki. Just one and a half years. Before that, she had lots of diagnostic experience in many other places, including Barbados, Grenada, Cambodia, I think too. Or was that just diving? Yeah. Okay. She got her bachelor's in biology with a minor in marine biology at Virginia Wesleyan University. And when she started talking to me about coming to the master's program, she was really interested in water quality and early life history stages of corals. And so that was great because that is exactly what her thesis is focused on. She's been an incredible graduate student contributing to so many different projects. She's an incredible field asset. She's also a Strongcoast Fellow. So on her committee is Dr. Maya Trotz, who's the principal investigator of the Strongcoast program, and a civil and environmental engineer at the University of South Florida. Also on her committee is Dr. Ashley Lillis, who was formerly with the Nature Conservancy. Now she's working with the Sound Ocean Science program. She's also a National Geographic Explorer. And she helped to guide Lindsay on some of the experiments that she did over on St. Croix. So I'm very excited about Lindsay's results. She's worked incredibly hard, basically doing a whole bunch of water quality collection and analysis where I was like, I don't know what we're doing. I'm glad that you can figure this out. So all of these results are really the result of tons of her time and effort. And also with contributions from the lab, so I'm excited to see lots of grant lab members here. And so I'm happy to turn this over in 1 second after I explain the process. So if you're not familiar with UBI defenses, Lindsay's going to give a presentation for about 30 to 40 minutes. And then we're going to turn it over for questions from the audience, probably first in the room and then on Zoom. And then we're going to close it down for questions from just the committee. So Maya and Ashley, if you guys want to hold your questions until that closed meeting, you're welcome to. You're also welcome to ask anything publicly if you want as well. So that's, we'll get started. Right. Let me.
Okay, everyone can hear me all right? Well, first of all, thank you all for being here today. A lot of you all helped with this project, so I'm really excited to share some of my findings with you guys. Sorry, just figuring out Zoom and presentation. So the title of my thesis is The Influence of Water Quality on the Early Life History of Caribbean scleractinian Corals. Now, coral reefs are one of the most iconic oceanic ecosystems. Unfortunately, we've seen significant losses in coral cover from things like bleaching and disease events, hurricanes, and pollution. And solutions to these global and regional problems are daunting, but when it comes to local stressors, particularly things like runoff and pollution, we do have the potential to take meaningful action as a community. So this is a map showing the Caribbean region with the locations of coral reefs highlighted in purple. You can see that the majority of reefs fringe small islands and are therefore highly influenced by land-based activities. The location of this study, the U.S. Virgin Islands, which is located just east of Puerto Rico. And the U.S. Virgin Islands consists of 3 major islands: St. Croix in the south, St. Thomas and St. John in the north. Now, some of our pollution problems here include things like runoff and wastewater. So sources of runoff and sedimentation can include things like clearing of vegetation, having a lot of unpaved roads. These are really amplified by our really steep topography here. So in these two photos, I have an example from Coral Bay on the top in St. John, and then the lower photo is Brewers Bay and St. Thomas. In both of these pictures, they were taken after rainfall events, and you can see this large sediment plume coming out into the bay. Now, both of these bays do have coral reefs in them and are in fact two of our coral monitoring sites. In addition to runoff, we might potentially have wastewater pollution entering our coastal waters as well. So we have point source pollution or potentially point source pollution, which would come from things like sewer line leaks and sewage bypasses. You can also have non-point source pollution, which would be caused by things like leaky septic systems. And this last one might be a particular concern for our territory. So in a report from DPNR, which is our Department of Planning and Natural Resources, they identified septic system failure as a major source of surface water contamination. So it's likely that we have high failure rates of septic systems in the territory. Additionally, we do have a city sewer system here, but it serves about 60% of the population. So that means 40% of our residents are primarily on septic. Now, wastewater does contain things like harmful bacteria, excess nutrients, particularly in the form of dissolved nitrogen, and solids, and these have all been shown to have detrimental effects to corals. Now, currently, there are no threshold values for these things on coral reefs. Water quality is particularly important for coral reefs because it has the potential to influence where reefs form, as well as how they handle and recover from stress. And this is known as resiliency. So resilient reefs mean that the coral population is able to reproduce successfully and withstand these less than ideal conditions. Corals, like many marine organisms, have a multi-phase life cycle. So corals can reproduce asexually. That's when pieces break off, form into a new colony. But in terms of sexual reproduction, that is especially beneficial because it allows for more genetic variation and greater dispersal. So in sexual reproduction, the two primary mechanisms are brooding and broadcast spawning. So this is a diagram of the reproductive cycle of a broadcast spawning coral. And spawning corals will release their gametes into the water column, which then fertilize and develop into free-swimming planula larvae. Breeding corals, on the other hand, will release competent larvae that are ready to settle. And another difference between breeding and spawning larvae, brooders contain symbionts from their parents, whereas spawning coral larvae don't obtain symbionts until after settlement. Now, once larvae settle and metamorphosis into a single polyp, we call them recruits. And then as these polyps multiply and grow, we'll call them juveniles until they reach sexual maturity. Now, throughout the cycle, there are potentially some major bottlenecks that can occur. So in kind of a keystone study, Arnold et al. 2010, he identified the three major bottlenecks that can limit successful coral recruitment to be the larval supply, their ability to settle, and then post-settlement survival. And there's evidence that water quality impairment can contribute to these bottlenecks. So in the larval stage, it's been observed that nutrients can reduce both motility and larval survival. However, many studies looking at this have seen mixed effects. So it does appear that these impacts of nutrients are specific to species, the level, and the type of the nutrient enrichment. Sediments can also reduce larval survivorship. Both sediments and nutrients can reduce settlements. Sedimentation has been shown to reduce post-settlement survivorship. And we also have this indirect impact of excess nutrients causing algal overgrowth, and that can be detrimental to young corals. In addition, nitrogen enrichment has been shown to reduce growth and calcification rates, which for small corals would be particularly detrimental. So the aims of this study: one, determine if there was an influence of water quality on coral reproduction, but also assess which phases in coral reproduction might be most sensitive to water quality. So this is a 2-chapter thesis. So the first chapter targets the larval and settlement phases. Well, chapter 2 focuses on settlement and post-settlement, like in juvenile stages. All right, so chapter 1, assessing Orbicilla annularis, larvae settlement under elevated nitrogen conditions. So our target species was Orbicilla annularis for this experiment. This is an endangered species of coral, and they are particularly important reef-building corals. Historically, Caribbean reefs were predominantly a lot of Orbicilla species. So these are particularly important corals to this region. They're also broadcast spawners and are target species for restoration. And in recent recruitment studies in Virgin Islands, we've observed low recruitment of the species. This has also been observed in the Florida Keys. So it's possible that Orbicilla species are struggling with their natural reproduction, and water quality could potentially be a factor in this. So we measured 4 variables in this experiment. So we hypothesized that larval survival, larval motility, settlement, and post-settlement survival of Orbicilla annularis would be lower with increased nitrogen. This experiment was done in St. Croix, partnered with the Nature Conservancy. So all larvae used in this experiment were Prussian larvae, came from reefs in St. Croix, and the experiment took place at the Nature Conservancy's land-based nursery. So for the sake of time, I am going to go through these experimental methods a little quickly. So please feel free to ask questions afterwards. Basically, to assess the effect of nitrogen enrichment, we use 4 different seawater treatments. So the first 2 treatments serve as control because no nitrogen additives were added to these waters. So our control was artificial seawater, meaning it was just freshwater, filtered freshwater mixed with instant ocean salts. And then our ambient seawater was seawater that came from a nearby lagoon that had been filtered in this lagoon. Now, we chose ammonium as an additive for our nutrient enrichment because it is a common component of wastewater. It's also exhibited very mixed effects on corals in previous studies. So we used an ammonium chloride powder, which was calculated to achieve 10 micromoles of ammonium in the low treatment and 20 micromoles of ammonium in the high. Now we did run into one issue in terms of sampling. We were not able to sample for dissolved nitrogen, which is what ammonium is. We could only sample for total nitrogen, so we could not ultimately confirm these ammonium levels. Okay, so there's 2 experiments to this study. So they're kind of 2 separate experiments. Experiment 1 specifically looked at larval survival and motility and larval settlement. So we used approximately 2-day-old larvae, 10 larvae per vial, which contained 40 milliliters of treatment water. And then we used 9 vials per treatment. These vials were submerged in raceways to maintain temperature. And this experiment lasted 7 days. Now, settlement plugs, which were preconditioned for about a month in ambient seawater, were added on day 3 and were left to settle for 4 days. Now, moving on to experiment 2. So this was a separate experiment done. We used settled recruits from the same parents, but that had not been previously exposed to these treatment waters. So all of these recruits had settled under ambient conditions. So settlement plugs with recruits were placed in 2-liter containers, again within raceways for temperature control. We observed their survival after 7 days. To look at our larval survival, we observed the larvae under a microscope and assessed survival on days 1, 3, and 7. So day 3 being when that settlement plug was added. For our post-settlement experiment, we just looked at survival on days 1 and 7. Now, we also looked at larval motility as a metric of competency. So these assessments were just done visually, and we did them twice a day during those 7 days. We categorized larval motility as swimming, being directional swimming. Probing is an activity that indicates they might be ready to settle, so they're kind of nosing for crawling around the bottom, around the substrate. Spinning would be non-directional swimming. So that means they're not doing as great, and then motionless, no movement, they're not doing so great. Let's see if this video will play. This is an example of a bunch of baby Orbicilla annularis. Most of the larvae in this video I would call swimming. So the results of our total nitrogen levels. So this is a time series of total nitrogen across both of the experiments. So just for the ease of visual ease, I've put both experiments up here. So the first 7 days are for the larval experiment, and then the day 7 to 14 are from that second experiment. You can see that our nitrogen levels did fluctuate somewhat, but we have this consistent order of total nitrogen where ambient had the lowest, then control, low, and high. Now, both control and ambient did have somewhat higher total nitrogen levels than we expected. And in particular, our control treatment ended up having higher nitrogen than the ambient treatment. So remember, ambient was the natural seawater. Control was just filtered seawater, filtered freshwater with salts. So ambient unexpectedly had lower, the lowest total nitrogen levels. And then in terms of statistics, we did see that in the post-settlement experiment, all treatments were significantly different, but in the larval experiment, only ambient and high treatments were significantly different from each other. Okay, so our motility results, we looked at the proportion of swimming and motionless larvae. These were not significantly different among the treatments. But in terms of probing behavior, this is a bar plot of the average proportion of larvae that we're probing in each treatment. The treatments are ordered from lowest nitrogen to highest nitrogen. Overall, this behavior wasn't observed that often, but we did see significantly more probing activity in the control treatment. In the for spinning behavior, again, this is average proportion of larvae that were spinning for each treatment. And we actually saw higher, significantly more spinning in the highest nitrogen treatment compared to control. So we can see from both of these figures that there is some different behavior going on in the control treatment. However, it doesn't seem to be an apparent trend in terms of total nitrogen levels. So our larval survival results, overall, pretty high survival, 58% across all treatments. However, there was no significant difference among these treatments. And then we had very low settlement. So actually only 4 settlers among all the treatments. And these were not, it was not significantly different. For our post-settlement experiment, so this is experiment 2, where we're looking at the recruit survivorship. This is a bar plot of the mean proportion of recruits that survived over those 7 days for each of the treatments. Again, they're ordered from lowest nitrogen to highest nitrogen. Overall, really high survival, 81%. However, no significant differences across the treatments. And one additional test we did run, because those settlement plugs that we used had different numbers of recruits on them. It was not consistent across each treatment container. So we did run a generalized linear model to look at the effects of treatment or recruit density on survival, and neither of these were significant. So to summarize chapter 1, we basically observed no influence of elevated nitrogen levels on either larval motility, survival, settlement, or post-settlement survival. It might be that these early stages of Orbicilla annularis are more resistant to nitrogen. It might be that they had little effect on these larvae because they were asymbiotic. So symbionts, not having symbionts may mean that they weren't absorbing this nitrogen as much. So results could vary if we use it. And again, these observations are only for the levels that we induced and the duration of the experiment. So it is possible that with a longer experiment, you would see some different results. So coming back to our hypothesis, we can reject that original hypothesis. Survival, motility, settlement, and post-settlement survival of Orbicella annularis were not lower with increased nitrogen. Okay, now we're coming from the lab into the field for our chapter 2, coral recruitment patterns in relation to water quality in the northern U.S. Virgin Islands. So in terms of water quality monitoring, our DPNR does sample for water quality for surface waters and for beaches. However, in the territory, nobody samples subsurface waters. So the main goal, or one of the goals of this study, was to characterize water quality at the reef level and identify potential impairment at our study sites in the U.S. Virgin Islands, the northern U.S. Virgin Islands. So we hypothesized that coral recruitment rates and juvenile densities would be lower at sites with poor water quality, and we're using nutrients, fecal indicator bacteria, and suspended solids as indicators of water quality. So there were 3 main components to this chapter. So we use data from the Territorial Coral Reef Monitoring, or TCRIM, which is an annual monitoring program that conducts benthic and fish surveys at fixed sites across the Virgin Islands. So we use data from the coral demographic surveys, specifically looking at corals less than or equal to 4 centimeters, which is how we identify or categorize juveniles. We also deployed settlement tiles to look at coral recruitment and analyze water quality. So this is a map of St. Thomas and St. John, where our study sites were located. We have 6 sites in St. Thomas and 2 in St. John, and each site is colored according to its expected impact level in terms of water quality. So sites that were further from shore, further away from human activity, we categorize as low expected impact. Black Point and Fish Bay. So these are, we classify these as medium. They are closer to shore, but they're in areas of the island that are less developed. And then Rupert's Rock, Megan's, and Coral Bay, they're colored darker red. We expected these to have a higher impact. They are closer to shore. They're also in areas of more human activity. And so we expected these might have some impairment. For our water quality sampling, so samples were taken by hand. Depending on the depth of these sites ranged from 5 to 20 meters. So we would either scuba or free dive and take the sample about a half meter above the benthos. We sampled for dissolved nutrients, which are dissolved inorganic nitrogen, or DIN, and then total nitrogen and total phosphorus. And from these, we calculated NP ratios. We also sampled for fecal indicator bacteria, so this included Enterococcus, E. coli, and total coliforms, and then total suspended salts, or TSS. We sampled in both the wet and dry season. So for each sampling event, we did 1 sampling event per month, and we sampled between 2 to 3 samples per sampling event. We sampled 3 months in the wet season and 3 months in the dry season. So 6 sampling events in total. And then our FIB and TSS were analyzed at UVI's Environmental Analysis Lab, and then nutrients were frozen and shipped to their respective laboratories. So for our recruitment deployment, so we use protocols from similar regional studies where we use terracotta tiles that are preconditioned. For our conditioning, we soak them under the UVI dock for a month, and what this does is allows a biofilm to accumulate, and they basically become a more attractive substrate for coral recruits. And then we deploy them to the reef just by screwing them into substrate, which is typically a dead coral. We leave the tiles on the reef for 6 months at a time, and we did 2 cycles of this. So we had a fall deployment and then a spring deployment from 2021 to 2022. So when we removed the tiles, we brought them back to the lab, visually analyzed them for recruits. So the bottom picture here is me using a magnifying glass to just scan and look for recruits. And then any recruits that are found, we photograph with a microscope camera. And that's the other bottom picture. As a cluster of recruits that were found, they're really small and just one polyp, so typically we identify them just down to the genes. And then for our coral juvenile assessments, so the T-crimp monitoring took place in the fall of 2021, but we supplemented this data with additional surveys in February 2022, where we just targeted juvenile corals, because not all sites were included in these demographic surveys. But basically for this protocol, a diver will lay out a transect and swim along that transect, everything within 1 meter. They identify, measure, and assess the health of the corals. Okay, so we measure a lot of water quality parameters. So to kind of make sense of all of these, we were inspired by a study done in Hawaii by et al. 2018. They created a water quality index to kind of rank their sites and understand if there was wastewater pollution happening. So the basic process of this was for each parameter, we categorize the data as low, medium, or high. And then we applied a weight to parameters that we felt were more important or reliable. And then what we basically get is one score for each site. So it allows us to combine all of this data. Okay, so this is what we used. So the parameters included were DIN, dissolved inorganic nitrogen, NP ratios, Enterococcus bacteria, E. coli, and TSS. Now, to establish these categories, we looked through the literature to determine what would be considered low, medium, or high. However, for some of these parameters, we did not feel there was sufficient data available, or that it didn't apply to our sampling and our data. So in those cases, we used data from our own study. So we used quartiles of our data. And then in terms of the weight, so we only weighted Enterococcus bacteria. This is a pretty commonly used fecal indicator bacteria. We also have threshold values for it from our own DPNR. So we were fairly confident in using this as a wastewater indicator. But keep in mind that all of these parameters can come from other things, not just wastewater. So the results from this index, applying it to our data, we now have a score for each of these parameters, and then a total score for each site. So these sites are now listed in order from lowest to highest score, or from least impacted to most impacted. And for the most part, what we found was that they did match our expected impact levels. So you'll notice I included the low, medium, and high from that map earlier. Typically, the sites that were offshore that we expected to be low did rank lower on the index, and then sites we expected to be high that were, closer to human development did score higher in this index. However, the one result that was a bit strange was Rupert's Rock. So looking again at a map of St. Thomas, this is a satellite map, so now we can see where areas of higher human development. And Rupert's Rock is located just outside of Charlotte-Amalie Harbor. So this watershed, we expect to have a lot of, things coming into it. It's also located right next to our cruise ship deck. So within like 500 yards of up to 3 cruise ships a day, we expected this site to show some type of contamination. However, it scored really low in our index, and it seems to be particularly low in terms of Enterococcus bacteria. So not a single sample from Rupert's Rock actually registered above detection for Enterococcus. In addition, the NP ratios are particularly low, and this seems to be driven by lower total nitrogen levels. So we're going to come back to this. Looking at our coral recruitment results. So this is a box plot of the average recruitment rates. So on the y-axis, we have recruits per day per meter, and then each site on the x-axis. Now, the line through each box is the median, while the red point is the mean. So these data were really zero inflated, meaning that we had a lot of tiles with no recruits on them. We did see significant differences across the sites, however. The letters above the box plots are from the post hoc comparisons. And in particular, Black Point had really high recruitment rates. We did also see pretty high recruitment at Dade and And looking at the species composition or genus composition, we saw by far dominated by aggressives. So in total, like 200 and so recruits, 211 of these were aggressives. Now, aggressive species are not, they don't tend to get super large. They're more of a plating or encrusting coral. They are brooding corals. We also sometimes call them a weedy coral because they're a little bit hardier than other species and tend to do well under stressful conditions. And then we saw a handful of parietic species. So again, these are smaller corals. Sometimes they're also referred to as weedy corals. What's interesting though about these findings is that all of the recruits found were brooders. So throughout both cycles of these tile deployments, we didn't find a single spawning coral recruit. Now for our juvenile densities, this is a bar plot of the average juveniles for each of the study sites. And we did see some significant differences between these sites, really high juvenile densities, specifically at Black Point and Coral Bay. Now, this is the same bar plot, but we've broken it out into species composition, or I should say genus composition. So the color represents which genus group they came from. You'll notice that across all of the sites, there's a lot of light blue for parietes. So across all the sites, it seems like parietes was the dominant genus. However, at Black Point and Coral Bay specifically, there were a lot of agarissids. So this Agaricida juveniles kind of seem to be driving those two sites having really high juvenile density. And then we did see some Siderastria species, that's in the light green there. Now Siderastria siderea, these do tend to get somewhat larger. They are considered important brooding corals. This genus is also a brooding coral. And then we did include Orbicella because, as we talked about in chapter 1, they're really important species to the region, but we hardly saw any Orbicella juveniles. So that's the dark green color. You can only see these slivers there. So basically both our recruitment and juvenile species composition was dominated by brooders. So now we want to combine these results with our water quality. So we performed a linear regression analysis, seeing if water quality, using water quality index scores as a predictor for recruitment rates. So this is a scatter plot where each point is a site, and we fit a regression model to it. You can see there's a slight positive relationship, but it is not significant. It's a very large confidence interval. We can also notice at this point, this kind of outlier point way up here is of course Black Point, our site with really high recruitment. So a little history about Black Point. Dr. Grant's lab has been doing recruitment studies across St. Thomas and St. John for a few years now. And they consistently find that Black Point is a recruitment hotspot. So this is a figure from a 2019 paper where she looked at coral recruitment in the presence of the sponge community. They found Black Point had the highest recruitment rate. Additional studies have taken place up until now. So basically between 2016 and 2022, previous master's student, Brad Arrington, saw really high recruitment at Black Point as well. And it's theorized that this could potentially be caused by hydrodynamics. So looking at the topography of Black Point, this is an outline of St. Thomas with Black Point circled in red. So Black Point is the little point that comes out up top, and then that big rectangle that's sticking out there is the runway. So what might be happening is coral larvae are kind of getting stuck in that area, and it's forming a sink for coral recruitment. So given this background about Black Point, we felt justified in removing Black Point from our regression analysis and running it again And now we actually have a significant positive relationship. So if you remember the hypothesis, this was not what we expected. What this actually means is that sites with higher fecal indicator bacteria, TSS, and nutrients actually have higher recruitment. We performed the same regression with those juvenile densities, so using water quality index scores as a predictor for juvenile densities. We did this analysis both with black point included and with black point removed. Now, there's a slight positive relationship, but neither were significant. So bringing these results back to our site map, now I have the points colored according to the water quality index score. So darker red points have higher scores, meaning more impacted. And then the size of the point is representative of the recruitment rate. So larger points have higher recruitment. And what you'll notice is that sites like Black Point, Ladies Bay, and Coral Bay that are located in these embayments tend to have both higher water quality scores and recruitment rates. Now, Fish Bay is also located in an embayment. To me, this site ranks right kind of in the middle in terms of both recruitment and water quality. But what's interesting about this, so for just some reference, we do have this pretty predominant east-west current across St. Thomas and St. John. So with these somewhat sheltered embayments, what might be happening is that currents are pushing both these water quality parameters and coral larvae into these areas, and they're creating a sink for both of these things. Now, this might also explain why Rupert's Rock was a bit of an anomaly. We expected it to have such much higher impact in terms of water quality, but it scored so low. And if you look at it on a map, it's not really in an embayment. So Charlotte and Molly Harbor is open and water is able to flow through there. So it's possible that there's enough water movement or flushing occurring that contaminants aren't able to accumulate. So to summarize some of our key findings from this chapter, we saw that both recruits and juveniles were dominated by brooding corals. Now, brooding corals or brooders tend to have high natal retention, which means that the babies typically settle close to the parents. Now, in addition, not really covered in this presentation, but we did a quick benthic analysis and noticed that Black Point had much higher composition of adult aggressive corals. So that's likely really contributing or also contributing to Black Point being a hotspot for recruitment. In addition, we saw that sites with high recruitment also had high water quality index scores. They were located in embayments. This leads us to postulate that recruitment rates were primarily driven by hydrodynamics and borehole supply. Now, hydrodynamics also appear to be important to reef level water quality, not just the proximity to human activity. So coming back to our hypothesis, because we saw that a positive relationship between coral recruitment and water quality index scores, and we saw no significant relationship between juveniles and water quality index scores, we can reject that original hypothesis. Sites with poor water quality did not have lower recruitment or juvenile densities. So now, kind of trying to bring both chapters together, overall, we observed no influence of water quality on coral larvae, settlement, or juveniles. And we have to think of early life stages within a population as being more sensitive to stressors, but these results might show that, in fact, these early life stages might be more resistant to water quality or poor water quality than we expected. Now, again, this relationship we observed was limited to the levels of water quality we saw at these locations, and of course, only the one species that we used in the experiment. Now, in terms of next steps, given that we saw so many brooding coral recruits and juveniles, it would be interesting to perform the larval experiment with the brooding coral larvae, especially given that brooding coral larvae do contain symbionts and might be more readily able to uptake dissolved nitrogen. Also, we talked about the importance of hydrodynamics, but the scope of this study did not include measuring water flow or currents. So future investigation into this would be really useful to really understand some of the trends we saw. And then lastly, we want to be transparent about our water quality index. So because we use our own data, some of our own data to characterize and rank the sites, the index was very relative. And in addition, we did compare our results to other studies, but found it very difficult given that reef level sampling is pretty rare. So overall, we could not confidently say that any of these sites were impaired since no threshold values exist for coral reef waters, nor can we confidently attribute any differences in water quality to human activity, aka wastewater. And then lastly, we do feel that these findings highlight the need for water quality thresholds. In addition, we could really benefit from establishing baselines of water quality, especially that we saw so much variation in water quality across regions, looking into the literature. So it might be that even just establishing baselines for your area could be really beneficial for future assessment. Given that, or even though we did not see a negative relationship between poor water quality and recruitment, given that they kind of coincided, like the high levels of these parameters did coincide with high recruitment, it does mean we should be careful in how we manage these areas. It might be that if in the future, if contamination does become a problem, these areas might be particularly sensitive, these sinks, if you will. And then lastly, we do think this information is particularly useful to watershed planning groups. They primarily look at land-based processes in these watershed assessments and watershed plans. But studies like this that look at benthic processes might be of interest to these groups. So I'd like to thank my funding sources. Thank you to my advisor, Dr. Brandt, and committee members, Dr. Lewis, Dr. Chops. Thanks to everyone in the program who helped with this project, and special thanks to Tasha and Brad. They were particularly helpful contributors to this project. And then I think we're ready for questions.